Good. So I'm happy to be here. I'll sort of give you an update on where we're at with our CELPPLUS project. And that's a project which is designed to improve the 2D culture for making better phenotypes for in vitro assays. And it's really sort of a technology project which we are working on. Let's see, how do I get this? So yes, it's a project which is a collaboration between Leo Pharma and then Orange University. We have two groups at Leo Pharma. One group at Leo Pharma, sorry, headed by Andreas Secinin, and then two groups at Orange University, one at Natural Sciences, my group in iNano, and then the Klaus Johansson's group up in clinical medicine in health, and then a number of postdocs and PhD students. So the project has got two main parts to it. The first is a generic technology development where we're trying to create some materials and platform that could be used to potentially improve the phenotype of cells for particular assays. And then a second part, which is to do with Leo Pharma, which is this demonstration of this technology to improve one particular assay. And the idea we have to improve our 2D culture and to give better assays is to work with giving signals from the surface of our materials in a controlled fashion, and to use those in addition to the soluble signals that you would normally add to be a better mimic for the sort of this in vivo environment, and that way to be able to give better signals and get better phenotypes. So what we're particularly interested in is using nanopatterns of proteins at the surface of culture wells in order to control how cells are able to form different types of adhesions. And in particular, we're interested for the case of the keratinocytes for Leo Pharma, working with hemidesmosomes, which are forming at the extracellular matrix, and also working with desmosomal adhesions, which are connecting two cells together. And the idea is that by controlling these through nanopatterns, we'll be able to control the signals given to cells and therefore their phenotype. So within the project, we are working with Leo Pharma, and they have an assay, which is an inflammation assay, looking with keratinocytes and looking at drug screening for reducing inflammation. And what we would like to do with the project is to use these nanopattern materials to be able to steer the primary keratinocytes into different phenotypes, which would then represent different layers of the skin, and then allow a more defined assay, which would give you sort of better readouts. So that's kind of the goal of the assay demonstration. So within the project, the first part is to do with the technology, and we've been working hard to create a method of creating protein nanopatterns on transparent substrates. We use a technique called colloidal lithography. In brief, we can take a glass substrate or other materials. We can deposit particles on the surface to create a pattern, and then through a different set of transfer steps, we can convert that pattern of particles into site-specific immobilization of polymers at a surface, which then can allow us to have both a stealth protein-rejecting background, but then we can have specific click chemistry reactions that we can go, and then we can use that to immobilize really whichever proteins we want into different patterns. And so this is sort of the base technology that we're working with. In this example, we've used, schematically, of course, we've used it to immobilize some cadherins into patterns, but of course it could be other proteins as well. And this is something we've worked with for a while at a research level, and we've shown that using these nanopatterns, we can, these are different nanopatterns of laminins, we can use those to control the way in which cells adhere to surfaces, and perhaps here we were showing that they could distinguish between forming focal adhesions or hemidesmosomes based on the pattern size. And so this is something we've done for a while at the lab level with sort of small scale. These are glass plates, but this is something which is not useful for Leo Pharma. In order to work, they need to work with much larger numbers of samples, more defined. So that's really what the project has been about, has been scaling up this type of technology in order to work not at a sort of a 1-inch glass plate that we've worked with before, but to work with a size that we can then integrate into a 96-well plate format, or potentially even a 384. So it was surprisingly difficult to do that. So it's in terms of scale up, everything is larger, things don't diffuse so well. So it's taken some time to work out a protocol to get our particles to stick. Using sonication, pressure cookers are an important step. Also having the right tools. So we do 3D printing of our own devices for drying and spreading liquid and so on. So has been successful. So this is an example of a patterned surface. This is SEM images, light microscopy images of 800 nanometer particles on a surface, and then this is one of these plates which is matched to the size of a 96 well plate. So we can make the particles to stick. We also had to then do this polymer mobilization, and again we were working a lot with sort of prototyping different devices that could help using 3D printing, but we could succeed with that. So we now have plates with patterned polymers on it. We need to integrate them into a 96 well plate format. We've been working with a commercially available sort of medical adhesive, laser cutting holes, aligning these, and we're able now to get these 96 well plates. And it's hard to use sort of hand things here, but I just wanted to show this, that we actually have real 96 well plates with nano patterns on them now. And so it's something that it was an important milestone for us. Using those, we then needed to show that we could turn them into protein patterns. So we did some work just using some model proteins with dyes on them. So we created a pattern on the surface which was having in some regions azide groups that could stick to a click chemistry reaction to DBCO. And so we had bovine serum albumin labeled with DBCO and a dye. And then we had other regions with biotin on it that we could use to immobilize streptavidin and in principle other proteins. So this was just a way of getting proof that we could make our patterns. And over our very large area, we can, this is a very zoomed in image. We've got these here 500 nanometer patterns, and we can create then a pattern of a protein in one location, or we can have two proteins. And this is something that we can do over a 96 well plate format. So that was sort of our generic part of our project, where we have this platform, and then we should show how that could be used in a particular application for Leo Pharma. And so in Leo Pharma's case, we were interested in using these nano patterns in terms of steering the phenotype of some primary keratinocytes that they've been working with into different phenotypes that could match to different layers of the skin. So that work is ongoing. To give you an idea of the type of experiments we're doing, we are working with a commercially available primary keratinocyte, the same ones that Leo Pharma are working with. And in that assay, you first expand the cells through 3 passages, and then you have this third passage keratinocytes, which are then purified a little and perhaps modified along the way. And these are the ones which are then used in the Leo Pharma assay. What we're interested in doing is to take those different cells at the different passages and to interact them with different pattern sizes. And so here we can create a plate where we have a number of wells with one pattern size or another pattern size of a particular protein. We can have different proteins in different wells. And then we can look into how the different cells are affected by our signals that we're giving it, and then look into the phenotype. And we're doing that through imaging, high-throughput imaging, screening imaging, and then we're setting up now PCR assay to look at specifics of each phenotype. So that work is ongoing. I just wanted to say something about how we're thinking about which proteins to use. One type of protein that would be relevant to use would be extracellular matrix proteins such as fibronectin and laminins, and they could be used to target different types of adhesions to the extracellular matrix, for example, hemidesmosomes or these focal adhesions. And in parallel, we're working together with membrane proteins that we're putting into nanopatterns, and there we're targeting these desmosomes, and that's through the different cadherin ligands. So the results are ongoing as experiments are on the way, but I just wanted to give you a feel for the type of thing that we're doing. So this is one example of a pilot study. So here we were comparing, we were using these membrane proteins. So we have the sort of the outside domain of different cadherins, which we can then click onto the surface oriented into patterns. And then in this example, we were comparing having that protein presented homogeneously across the surface or in the nanopattern. And we have this hypothesis, which we've shown in other cases, works where the size of the pattern can be used to steer how the cells are forming adhesions, and then the hypothesis is that will affect differentiation. So here we see basically the same adhesion of the cells to all the different surfaces, but what we do is see differences in differentiation. Here we have a marker called involucrin, which is looking at differentiation of the keratinocytes. And for the homogeneous surfaces, it doesn't make so much difference which proteins you put down, you get adhesion. But here we're showing that depending upon which of the cadherins we have, we see differences. And this is clearly a pilot study, but it's something which is encouraging. And then we'll be looking into how the formation of these adhesions correlates with the differentiation. And so we're using imaging. We can look both at the cell level, but we can look down at the adhesion level and see what adhesions are forming. So this is an example of using a pattern of cadherin, and then looking at the formation of desmosomes by looking for these DSCG2 and DSC2, and then also whether it's a mature or not by looking at other aspects of this adhesion. Yes, so that's sort of the first part of the project. The generic part is complete. We're now working on steering this differentiation of the keratinocytes to develop different phenotypes. And then next year we'll be working together with Lea Pharma to use those keratinocytes to show that we can have a more defined and better screening assay. Yeah, we've been asked to comment a little bit about the collaborations and how the open science works. And one thing, a comment that I wanted to make was that, when I first came into this, I sort of rather naively perhaps thought that it would be just, it would be open and it would be free and we could, discuss whatever we wanted, come up with great ideas. But I realized that that's not the case. And it's not because this project is not open, but because all of the surroundings are still closed. And what I mean by that is that when we work in an ordinary project, a closed project, we can brainstorm, we can have good ideas, we might come up with things which are potentially relevant for our project, but we test them and maybe they don't work. But those things could be very relevant for other projects, and you can protect it and then do IPR, and you can then use those in future projects. And so something that sort of is an issue here is that if you come up with ideas within the project, which maybe are not useful for this project, then it's not something you're then able to protect and put under IPR. So one has to be a little bit careful thinking about what methods and materials and types of ways one should try, in that they should really have a good chance of being applicable in this case, because you don't want to somehow ruin the possibility to use it in other application areas. But on the other hand, it's fantastic for being able to talk about the things that we're doing with other people, making connections, and that way is very good. Yeah, so then this sort of idea of something to do with the connection to the closed projects of the future. In our application, we are developing a sort of a generic technology, so it's clear that it could be very useful and applied to other areas. So this is sort of broad potential use. It could be working in assays in basically all disease states where it's something that you could improve at a 2D level. And for us, we have been including this technology into applications for closed scientific projects already. For the closed innovation projects is something we'd like to do in the future, but it'll really require demonstration of this use case together with Lea Pharma. So that's something that will be coming next year. So with that, I'll stop with this slide, which is just showing our progress.